Welcome to Lecture 16 of Biology 116, entitled Development 1. In the previous lectures, we've looked at the reproductive systems of both the male and female human anatomy. And now what we're going to do is discuss the culmination, the climax, and the eventual development of the individual after the necessary reproductive events that we've covered. Those reproductive events included things like spermatogenesis and oogenesis. It included things like the delivery of sperm and the retrieval of sperm, and it will culminate in this idea of fertilization, which will follow with development. That's our path of knowledge thus far, and that's what we're going to sort of capitalize on in the next two lectures, which are Development 1 and Development 2. So in order to begin, we'll entitle this first flowchart on development as a simple introduction. And here we're going to sort of hash out what it really means, this term of development. And so when we think of this, many of us have seen this word before. All of us have basically seen this word before. But in terms of biology, what do we mean by development? Biologically, when we say development, what we're saying is the following. Development is full of changes. It includes all changes that occur, and these changes are going to occur during the entire life cycle. During entire life cycle of the individual organism that's in question. Of individual, we'll just say. So, the big thing here is that we're going to be seeing many changes over a long period of time, and the starting point of all this is the moment fertilization happens. And that's what's going to be highlighted a lot in this lecture. What happens post-fertilization and even pre-fertilization? How do we get to that point and begin the changes that occur during the entire life cycle? Because remember, we all start as one zygote and eventually become the incredibly advanced organisms that we are today, watching this video, listening to me speak. So let's go over that. In addition, development is really a science that's based off of model organisms. It's very difficult to understand development without model organisms because these are going to be specific species, many different species, that are going to be chosen for this type of research. Now, these are chosen specifically because each species that is, let's say, a developmental model organism that a scientist wants to use, they're very easy to do the following two things. These species are easy to both study, and we'll see why later on, and also to maintain. It is a little bit difficult to study development in humans because, first of all, we cannot force development onto humans. We cannot force humans to procreate. And it's also difficult to study this internal sort of development that humans have. So therefore, we're going to capitalize on other model organisms where it's easier to study development and easier to maintain those organisms. Those organisms include, but are not limited to, things like sea urchins, which are echinodermatas, and we'll get into these a little bit more as we move forward. We'll also talk about the frog development. We'll also talk a bit about chick development, generally birds here, and also nematodes. Nematodes are an incredibly great model organism to look at things like development because they're very easily studyable and maintainable, if those are words. Again, each presents their own advantageous characteristics that are good uses as models for development. Difficult to do in humans, easier to do in this, as we'll see. Sea urchins are going to be big as we move forward. So that's a basic overview of development. Lots of changes are occurring throughout the lifetime of an individual, whether it's a sea urchin, frog, a human, chick, nematode, you name it. That's development. Now, in order to begin development, you have to first be sort of complete a process known as fertilization. And remember, this is the climax, the culmination of the male reproductive system and the female reproductive system, of spermatogenesis meeting oogenesis and creating a zygote. How does that happen? Fertilization, of course, is just going to broadly be referred to as sperm meeting egg, sperm plus egg, or ovum. And this is going to be the classic scenario where we have a sperm that has a flagellum and is relatively small, meeting up with an egg that is larger and also that's going to be non-motile. Now, we are going to see in great detail why the egg is larger, why the egg is non-motile, and why the sperm is smaller, flagellated, and motile. as we move forward in this lecture. Overall, the culmination of this meeting between sperm and egg can broadly be referred to as fertilization that ends up and concludes in the development of a zygote. The zygote is essentially the fertilized egg, as we've said many times before throughout our biological studies. And this fertilized egg, it's important to remember, is a single cell, a combination of mom's genes and dad's genes that's going to eventually develop into the organism that is born, and then that develops post-birth. So this is the main event that sort of starts everything. And so we have to talk about this in a lot more detail as we move forward, and that's going to be a big point of this lecture. How do we have this specific zygote formation with the specific events associated with it? In addition, fertilization causes some consequences. And when we say consequences, we usually have a negative connotation, but in terms of a biological understanding, consequences are just results of fertilization. And those results include things like restore the diploid number. Now, why are we restoring this diploid number? How are we restoring it? Remember, a zygote is officially 2N, whereas a sperm is haploid, it is N, and an egg is also haploid, it is N. That combination, which we'll term and sort of hash out in greater detail, is the idea of restoring the diploid number. And this is supposed to say restore. There we go. Now, in addition, usually fertilization in most species, this moment actually also determines the sex of the individual. We're not going to get into the details of the offspring specifically of this event, but that's another major consequence that you have to understand. And this is something we're going to talk about a lot, the activation of the egg. When sperm meets egg, the egg turns into this activated state. This is going to stimulate a variety of reactions, which we'll see. Those reactions will be necessary for a variety of reasons, which we'll see. And then overall, this egg activation and reaction stimulation will sort of push forward and begin development. Development begins as a result of this. So this is our basic overview of the scope of this lecture. We're going to look at lots of changes occurring post-fertilization and even pre-fertilization at the sperm and egg level, and then hopefully begin to look at a more macro level of development in the next lecture. So right now this is our basis, and we'll move forward from this in the next couple of flowcharts and videos.